Hi everyone, this is chapter 12 on characterizing and classifying eukaryotes. So in microbiology, we mostly study prokaryotic cells, which was the topic of our last chapter and the topic of a lot of things that we talk about in here. But there are a few eukaryotic cells that we talk about, like fungi, protozoa, helminths, things like that, that are technically considered to be eukaryotes. So we need to cover them in here. Um, so that's what this chapter. So here are a list of the eukaryotic microorganisms that we'll be talking about a little bit here. Protozoa, fungi, and algae, slime molds, and water molds. The last three don't really apply to medical microbiology very much, but we're going to briefly cover them so that we can say that we did. But we're more interested in the protozoa and fungi in medical microbiology. These include both human pathogens and organisms that are vital for human life. So how do eukaryotic cells reproduce? Well, it's a whole lot more complicated than what we just saw with prokaryotes. Prokaryotes, it was really straightforward. You just make a copy of your single chromosome and you split yourself in half, or you bud, or you fragment. But however you do it, it's a really simple process. Most eukaryotic cells have the DNA packaged as chromosomes, and there are more than one, and that is in a nucleus, and that is surrounded by a nuclear membrane. So there's a variety of methods of asexual reproduction, meaning that you do not need the contributions of a second individual. Many reproduce sexually by forming gametes and zygotes. Algae, fungi, and some protozoan reproduce both sexually and asexually to make it even more complicated for us. Nuclear division. This is when the nucleus has one or two complete copies of the genome. When we have a single copy, we refer to the nucleus as being haploid. Most fungi, algae, and some protozoan are considered haploid. If you have two copies of every gene, you're considered to be diploid. So plants, animals, fungi, algae, and protozoan, if they have two copies, are all diploid. There's two main types of reproduction that we talk about. There's mitosis and meiosis. So mitosis is when the cell partitions itself after replicating the DNA equally between two nuclei. This maintains the ploidy, or the number of chromosomes, of the parental nuclei. There's 4 phases in this that you probably learned about in another biology class. They were prophase, metaphase, anaphase, and telophase, or PMAT. All right, a quick diagram of mitosis where you had a cell that was diploid. It had copied its DNA, and then it went into the mitotic phase where it went through prophase, metaphase, anaphase, and telophase. And at the end, we ended up with 2 separate cells, and each of those cells had a full complement of the parental chromosomes. Another version of what we talk about in eukaryotic cells is called meiosis. So meiosis is how we produce gametes. So this was nuclear division that partitioned the chromatids into 4 nuclei rather than ultimately making 2. And each of the nuclei, or the new cells that were being made with a nuclei, were going to be a haploid. They had half of the original material. There were 2 stages in this, and that was meiosis 1 and meiosis 2. So mitosis makes a clone. You make an exact copy of the parental cell. Don't need another contribution of DNA from a secondary source. You just literally make a copy of the genetic material, and then you go through this process of PMAT to split yourself in 2. Meiosis is how we're going to make cells that ultimately are going to join together to produce a new individual. And so in meiosis, we're going to start with a diploid cell and we're going to reduce it down to a haploid cell. Sometimes they call meiosis reduction division for that reason. So whenever you're looking at these 2 situations, the phases or the stages are the same: prophase, metaphase, anaphase, telophase. The difference is in mitosis, it'll just be prophase, metaphase, anaphase, telophase. If it's meiosis, then it'll be prophase 1 or prophase 2, metaphase 1 or metaphase 2. It'll have a Roman numeral behind it. And that's what tells you that it is a meiosis question rather than a mitosis question. So in meiosis, you actually go through 2 rounds of PMAT, and in between those 2 rounds, you don't replicate the genetic material, and what you end up with is, so this is showing you metaphase 1, meiosis 1, and then this is showing you what happens when you have meiosis 2 happen. So we end up with 4 cells that have half the chromosomal material, because the idea is, if we were just talking about mammals, for an example, we would have a sperm and an egg, and those two would join together to produce a zygote, and we would be back to the original chromosomal number. If our result of meiosis had a full complement of chromosomes in certain living things, that would be too many when they join together. So let me just use the example of humans. We have 23 pair of chromosomes. 23 you got from mom, 23 you got from dad. 23 came from the egg, 23 came from the sperm. When we put those together, we made 23 pairs of chromosomes. If each sperm had 23 pairs of chromosomes and each egg had 23 pairs of chromosomes, we'd have 46 pairs of chromosomes, which is 92 chromosomes, and that's way too many. All right, so that's the whole idea of meiosis, is that we're going to reduce the chromosomal number down so that we can rejoin with a secondary source and go back to the original number. So here's a nice chart that sort of summarizes all of the stuff I was saying, mitosis and meiosis. And so it talks about the DNA replication, how that happens. The phases, again, prophase, metaphase, anaphase, telophase, or PMAT are the same. The difference, and you can see this real clearly, is in mitosis, there's no Roman numeral after the phase, but when we're talking about meiosis, there is always a Roman numeral, either a 1 or a 2. The formation of tetrads does not occur in mitosis because we never have 4 copies. In early prophase 1 of meiosis, we actually have 4 copies of the chromosomes. Crossing over doesn't occur. Crossing over is how we get some new traits. We scramble up the traits that way. Crossing over does occur in meiosis. That's why you and your brother have both the same parent, parents, and you both have different traits. You look different because of this whole idea of crossing over. The number of accompanying cytoplasmic divisions that may occur, of course, in mitosis, it's just 1. In meiosis, there are 2, and the resulting nuclei is that with mitosis, we have 2 nuclei that are the same ploidy or have the same number of chromosomes as the original, and in meiosis, we have 4 nuclei with half the number of chromosomes from the original. So that's how we divide all of that chromosomal material. Then we have to talk about how the cell actually splits itself in two, and that process is called cytokinesis. So cyto means cell, kinesis means to move. So this is occurring simultaneously with telophase in mitosis. In some algae and fungi, it's postponed or does not occur at all, and so you can end up with multinucleated cells in that particular case. So there's a lot of different ways for cells to go through this division process. It depends if you have a cell wall or not. If you have a cell wall, you have to build a division between a wall between you and the new cell that's being made. If you don't, and you're like an animal cell, you can simply squeeze yourself into 2. There's a lot of ways for it to happen. At the end of the day, what we want to do is we want to separate the 2 new cells that we've made, or the cells we've made, from each other so that we have 2 new cells. A little bit different way to do things is gyogeny. This is not something humans do. In this case, we have multiple mitosis that happens over and over and over, and then we split them all at once. It's like we're doing a batch mitosis rather than doing it just creating two cells each time. We're going to create a whole bunch of them and then we're going to split. So that's kind of something different. Again, humans don't do this, but other animals and other uh, creatures do. What about the classification of eukaryote organisms? Early taxonomy schemes grouped organisms together based on what we could see, what was structurally similar. Modern classification is based more on nucleotide sequences. So when we take the DNA from animals, we can, or any living organism, we can look at similarities and we can figure out their evolutionary relatedness. And so it's just showing you how it's changed. So this has really changed a bunch. So when I went to college, this was not a thing because I mean they knew about DNA, but they weren't sequencing DNA or doing any of that kind of stuff. And when I went to graduate school, we had moved up to that level. So I learned it a very different way than the way you learn it now because there wasn't any DNA to analyze back when I went to college. But you can see things have really, really changed between the late 20th century, which was when I went to college, and the 21st century, which is where we are now, and how we're able to divide things into much more definitive groups based on their sequences of DNA that you and I can't see without all of the analysis. So let's talk about some of these eukaryotic things here. Let's talk about protozoa. This is a diverse group that's defined by three characteristics. Number one, they are eukaryotic. Of course, eukaryotic, and I guess I should have defined this in the beginning. Eukaryotic cells, remember, have a compartmentalized nucleus, and they have a whole lot more of those fancy organelles that I talked about in a previous chapter. They're a much more complicated cell. Protozoan are unicellular though, so they are just a single cell doing all the stuff, and they lack a cell wall. They are motile by means of cilia, flagella, or pseudopods, or they can have combinations of those. There is one subgroup that doesn't move, and that's the apicomplexians. So the distribution of protozoa, they require moist environments. So most of them are going to live in ponds or streams or lakes or oceans. Most of them are plankton. So if you're familiar with SpongeBob and you know who plankton is, plankton was a protozoan. Others live in moist soil, beach sand, or decaying organic matter, and very few of these are pathogenic. There are a few, but not a lot of them. Their morphology or their shape, this group has the greatest morphology of all of the groups that we look at. Some actually have two nuclei. They have a macronucleus that has many copies of the genome, and then they have a micronucleus. They may vary in number and kinds of mitochondria. Some of them have contractile vacuoles, which help them to pump water out of the cell. So as water builds up in the cell, they have like these it's like a balloon and they squeeze it and all the water jets out of it. And they have different stages of life cycles. There's a motile feeding stage that is called a trophozoite. There's a resting stage that's called a cyst. So there's the contractile vacuoles. They always look like a sea star. They always have those kind of arm-looking things on there. Basically, they're siphoning water in to keep all of the osmolarity inside of the cell the same, and then when they get full enough, they just jet that out. What about their nutrition? Most of them are chemoheterotrophic. They obtain their nutrients by phagocytosing bacteria, decaying organic matter, other protozoa, or the tissues of their host. A few of them absorb nutrients from the surrounding water. The dinoflagellates and the euglenoids are photoautotrophic, meaning they can make their own food. I believe plankton was a photosynthetic organism. He was green. Most of them reproduce only asexually. There's a few that go through binary fission or schizogeny. Few also have sexual reproduction. Some become gametocytes that fuse to form a diploid zygotes, and some utilize a process called conjugation. Our classification of protozoa has shifted dramatically over the years. It's been revised and updated based on nucleotide sequences. The current scheme, which will probably change again, has the protozoa in 6 groups: the Parabasala, the Diplomonodidia, the Euglenozoa, the alveolites, the Rhizaria, and the Amoebozoa. Boy, those are something to say tonight. So a little information about each of these. The Parabasala, these lack mitochondria. They have a single nucleus. They contain a Golgi body-like structure that's called a parabasal body. And the important one in medical microbiology is Trichomonas. All right, so looking under a scan electron micrograph, this is Trichomonas vaginalis, which is a sexually transmitted infection. All right, our next group to look at is Diplomonadida. These also lack mitochondria. They have mitosomes in the cytoplasm. The mitochondrial genes are found in the nuclear chromosomes. These also lack Golgi bodies and they lack peroxisomes. They have 2 equal-sized nuclei and multiple flagella. They have a prominent diplomonad, and the one of interest to us is Giardia. Giardia is the one that people refer to as backpacker's diarrhea. You can get it from drinking water like a freshwater stream or a mountain stream or something like that. It often happens to is infected people when they go up into the mountains and they go hiking and they think, oh, the water looks so clean, I'll just drink it, and they end up with Giardia. All right, the Euglenozoa. These are characteristics of both plants and animals. So they are, for a long time, these were considered to be animals, and then they were considered to be plants, and then they're considered to be protozoa, and now they have characteristics of both, so they're kind of in that weird place in between. The flagella contains a crystalline rod of unknown function. The mitochondria have disc-shaped cristae, and there's 2 groups, the euglenids and the kinetoplastids. The euglenids, these are photoautotrophic unicellular microbes with chloroplasts. They are, as I said, they were historically classified as plants. They store food as polysaccharide called paramylon. The kinetoplastids have a region of mitochondrial DNA called a kinetoplast. Some of the kinetoplastids are pathogenic, including trypanosoma and leishmania. So here are 2 representatives of the kingdom of Euglenozoa. The alveolites, these have a membrane-bound cavity called an alveoli, and the purpose of that is completely unknown. These are divided into 3 subgroups, the ciliates, the apicomplexans, and the dinoflagellates. So taking a look at the membrane alveoli that are found in some protozoa. So look for those kind of open areas. There's one up at the top. They've got it pointed to a couple of them. The ciliates use cilia to move themselves in water. This is the one that always, to me, they look like they have a little fuzzy, furry border on the outside of them, and they move those cilia, and it helps them to. All of them are chemoheterotrophs and have 2 nuclei. Balantidium is the only ciliate that's pathogenic to humans. The apicomplexans, these are chemoheterotrophic pathogens of animals. They have complex organelles, allow them to penetrate into host cells. So Plasmodium, Cryptosporidium, and Toxoplasma all cause diseases in humans. And this is Didinium. This is a predatory ciliate. So he's on the left, and he's devouring a paramecium. So you don't think about that kind of stuff, predation going on in the microscopic world, but it surely does. All right, a little bit about the dinoflagellates. These are unicellular microbes with photosynthetic pigments. These were historically classified as algae. So plankton on SpongeBob, he is a dinoflagellate. A large portion of our freshwater and marine plankton are dinoflagellates. The motile dinoflagellates have two flagella. Many of them are bioluminescent, so they glow. There is an abundance of them in marine water that cause a disease called red tide when too many of them bloom at the same time or multiply. And some dinoflagellates are able to produce neurotoxins. So here is a motile armored dinoflagellate. Looks like something out of the Age of Dinosaurs. The Rhizaria, these are amoeba that are protozoa that move and feed with pseudopods. The amoeba exhibit very little uniformity. The Rhizaria are amoeba with thread-like pseudopods. The foraminifera, these often live attached to the ocean floor. Most of these are fossil species. And the Radiolaria, these have ornate shells of silica and live as part of the marine plankton. So there's some pictures of them. Again, they don't cause any diseases, so we just casually go by them. Radiolarians are actually really pretty. The amoeba, these have long-shaped lobe-shaped pseudopods with no shells, and they include some important human pathogens, including Naegleria, Acanthamoeba, and Enteramoeba. Slime molds are now classified as Amoebozoa. There's two types: the plasmodial slime molds and the cellular slime molds. So taking a look here, this is a review chart where you have each of the groups that we talked about, and then the representative examples, and then page 2 of that. That allows us to move along to fungi. So fungi are chemoheterotrophic. They have a cell wall, but it is made of a different substance, and that substance is called chitin. They do not perform photosynthesis because they don't have any chlorophyll, and they are related to animals. What is their significance? They decompose dead organisms and recycle their nutrients. They help plants absorb water and minerals. They can be used for food. They can be used in religious ceremonies. They are used to manufacture foods and beverages. They produce antibiotics and other drugs. They serve as an important research tool. 30% of them cause diseases in plants, animals, and humans, and they can spoil fruit, pickles, jams, and jellies. There's two basic body shapes, and we talk about the morphology. Molds, composed of long filaments that are called hyphae, and yeasts, which are small, globular, and composed of a single cell. Some fungi are dimorphic. They produce both yeast-like and mold-like shapes, and they change in response to their environmental condition between those two. So looking at the fungal morphology. So we're looking at hyphae up there. Hyphae look like roots. They're not roots, but they look like it. They're long, slender projections. And then yeast-like cell or yeast cells down at the bottom. So there is a fungal mycelium. When we put all of those together, you see all of that net kind of fibrous type stuff. That's considered to be the mycelium. So fungi are going to acquire their nutrients by absorption, and most of them are saprobes. Some of them trap and kill microscopic soil-dwelling nematodes. Hostoria allows some fungi to derive nutrients from living plants and animals. Most fungi are aerobic, and many yeasts are facultative anaerobes. So here is a fungus that is predatory. and it's predating a nematode, which is a worm, and it is going to kill it. So how do fungi reproduce? They all have some means of asexual reproduction involving mitosis and cytokinesis that we talked about briefly already, and most also reproduce sexually. They can do this through budding and asexual spore formation. Yeast bud in a manner that's similar to what we saw with our prokaryotic budding. Some yeasts produce long filaments called a pseudohypha. The filamentous fungi produce lightweight spores that disperse over large distances. That's what a lot of the things that you see out in nature, what we technically call a mushroom, they let the air carry their spores wherever they need to go. The asexual spores of molds are grouped by mode of development. So looking at some asexual spores of mold. The sexual spore formation, the fungal mating types are designated as plus and minus, and there's 4 basic steps. And there are those 4 basic steps. So if you start at number 1, which is in the red box, we had a dikaryotic stage. So we had 2 copies of the genetic material, 2 nuclei. And then we proceed into the diploid stage. The nuclei are going to fuse, and then we are going to go through mitosis or meiosis, and then we're going to go into the haploid stage, and then we're going to go through mitosis and cell division, and the tips are going to fuse. So it's real complicated. All right, so looking at classification of fungi. So fungi use the term division instead of phylum. And that being said, some books will still use the term phylum, but to a mycologist, in the purest sense, we use the term division. So we have the division Zygomycota, the division of Ascomycota, the division of Basidiomycota, and the Deuteromycetes. Anytime you see my, you know it has something to do with fungi. So a little bit about the mycota. 1,100 known species. Most of them are saprobes. Others are obligate parasites of insects and other fungi. They reproduce asexually via sporangiospores. They produce microsporidia. These were once classified as protozoan. They are more similar to Zygomycetes by genetic analysis. And the microsporidia are obligate intracellular parasites. They spread as small resistant spores. So there is sporangium, sorry. The Ascomycota, there's 32,000 known species of this. These form ascospores in sacs that are called asci. They reproduce asexually by conidiospores. Includes most of the fungi that spoil our food. Some infect plants and humans, and many are beneficial, including penicillin, which is a fungi, and Saccharomyces. So these are some ascocarps, fruiting bodies of the common morel, which people like to get to eat. So you might have seen these before. The Basidiomycota, 22,000 known species. Mushrooms and other fruiting bodies of Basidiomycetes are called basidiocarps. The Basidiomycetes affect humans in several ways. Most of them are decomposers that return nutrients to the soil. Many mushrooms produce toxins or hallucinatory chemicals. This is what they talk about with magic mushrooms. And some cause extensive, expensive crop damage. So the fruiting body is the part that you see above the ground that's pretty, right? Or looks like what we think of as a mushroom. So these are mushrooms. If they are poisonous, they're called toadstools. The Deuteromycetes are a heterogeneous collection of fungi with unknown sexual stages. So this is the Deuteromycotes are kind of interesting. So there's not a lot of people who study fungi in the world. I know that's an amazing thought, but there's just not a lot of people who study this. And so when they find a new fungi, they immediately put it in Deuteromycetes. In other words, we don't know what you are. And sometimes they've sat there for a very long time, like decades, before someone actually was able to determine where they actually what they belong, where in those other groups that we were talking about, those other divisions where they belong. Most of the Deuteromycetes belong to the division of Ascomycota based on their rRNA analysis. They no longer consider this a formal taxonomy group, but it is where they put them when they first discovered them because we don't know where else to put them. Until it used to be until they saw the sexual stage, they didn't know if it was Basidiomycetes, Ascomycetes. They didn't know where it belonged. And sometimes you just didn't have anybody around that was observing them, right? And so they would stay for a long time. Now that we have the ability to do DNA analysis, we've pretty much eliminated this division, but it's still out there as like a temporary holding spot for, hey, we don't know where you belong. So you want to be really, really careful with fungi. So fungi, technically all fungi are edible. Some of them are only edible once. So a lot of fungi are poisonous. And so you never want to just collect mushrooms unless you really know what you're doing. And because there's some of them that look so very similar to each other, and one of them is poisonous, can make you very, very sick, maybe even cause your death, and another one is delicious. So you want to make sure if you decide to go foraging that you really and truly have someone who knows that information really, really well. Even as much as I know, which is not enough about fungi, I would absolutely not eat a mushroom that I found in the wild because you just never know. That lets us talk about lichens for a minute. So lichens are a partnership between a fungi and a photosynthetic microbe. So remember, fungi can't make their own food, but if you hang out with something that can, then the two of you can both have a beneficial relationship. The fungus is going to provide nutrients and water and protection, and the photosynthetic microbe is going to provide carbohydrates and oxygen. So they're going to have a commensalistic relationship where they're both benefiting, right? These are abundant throughout the world. I have lichens on every tree in my on out on my farm, and I've got 10 acres of trees out there. They grow everywhere. They're those kind of gray, crusty looking things. They grow in almost every habitat. There's three different shapes to this: the foliose, the crustose, the fructose. Um, they create soil from weathered rock. So when you have an area that has been completely decimated, down to the rock, like you had a volcanic explosion of some sort, and everything was just returned to the point where it's just rock, the first thing that happens is that lichens come in and they start to degrade those rocks and produce soil. And once you produce the soil, then you can have plants, right? Some lichens provide nitrogen in nutrient poor environments, and a lot of animals eat them because they, I guess, are like, I don't know, little algae potato chips because I see squirrels eating them sometimes too. So you've probably seen lots of uh, lichen out there in the world if you just look at a tree or a rock. There's what they look like, the gross morphology. So the foliose ones look kind of like a leaf. The crustose ones look like crunchy stuff, which is what I see the most of down here in Georgia. I don't know what it's like where you are, but that's mostly what I see here. And the fructose are the ones that kind of look like spiky little leaves. So some tables to, or a table to look at the characteristics of our fungi, the division and type of sexual spores. That is how these guys are organized. Zygomycota, Ascomycota, Basidiomycota. Their sexual spores are specific to that division. Their distinguishing features and their representative genera. The one that's on here that I want to talk about real quick is Rhizopus. Rhizopus is black bread mold. You guys have all seen this before when you buy a loaf of bread and you let it sit for too long and it gets the black and green kind of fuzzy stuff growing on it. That's Rhizopus. All right, moving along to the groups that typically don't cause any human diseases, but just to be thorough in this group, in this course, we'll cover these. Algae are simple eukaryotic photoautotrophs. They have sexual reproductive structures in which every cell becomes a gamete. They differ widely in their distribution, morphology, reproduction, and biochemical traits. Most of them are aquatic. They live in the photic zone of fresh, brackish, and saltwater bodies. Brackish is water that's both salt and fresh water. We have a lot of brackish water where I live. Um, they have accessory photosynthetic pigments that trap energy of short wavelength light and allows the algae to inhibit deep parts of the photic zone. They can have a lot of different shapes, unicellular, colonial, simple multicellular bodies. Marine algae can have holdfasts that function as anchors so that they don't get washed off. Reproduction in the unicellular algae, you have asexual reproduction that involves mitosis followed by cytokinesis. In sexual reproduction, the individual gametes form zygotes that undergo meiosis. Reproduction in the multicellular algae, they reproduce asexually by fragmentation and they reproduce sexually by alternation of generations. So alternation is generation is not something that we cover in this course. If you take a botany course, you definitely, or even a gen bio course, you'll talk about this. But it is the idea that you go back and forth between being diploid and haploid. In plants, a lot of plants do this, and algae are precursors to plants. How do we classify them? The classification of this is very unsettled as well. Schemes that are based on different features, including the different types of photosynthetic pigments that they possess, the storage products that they make, their cell wall composition. There are various groups, the division Chlorophyta, the kingdom Rhodophyta, Phaeophyta, Chrysophyta. There's a lot of groups here. The chlorophyta are what you and I think of as green algae. They have numerous characteristics that they share with plants. They have chlorophylls A and B. They use starch and sugar as their food reservoirs, or as their reserves. They may have cell walls made of cellulose, and then they have what makes them put them in this division is that they have similar 18S ribosomal RNA sequences. Most of them are unicellular and filamentous and live in fresh water. Some are multicellular and they grow in marine waters. The kingdom of Rhodophyta is red algae. These have a red accessory pigment. They use the storage molecule glycogen. Their cell walls are composed of agar, which is why I wanted to talk about them, because agar is what we use to make our petri dish, the substance we grow our microbes on in our petri dishes and our test tubes. Both are, and carrageenan. These are both used as thickening agents, mostly in things that you wouldn't be you'd be very surprised, like ice cream. Non-motile male gametes are called spermatotia, and most are marine algae. So there's what they look like. The Phaeophyta are brown algae. These are their motile gametes have two flagella. They produce chlorophyll A and C, keratin, and xanthophylls. Most of them are marine algae. They use lamarian and oils as food reservoir. Their cell walls are composed of cellulose and alginic acid. Alginic acid is used medically and as a thickening agent. They also have a hairy flagellum. Here is a brown algae giant kelp. So if you have ever been to California or you're from California, they have big uh, acres and acres of this giant kelp out there in the ocean. And this is what it looks like. The Chrysophyta, these are golden algae, yellow-green algae, and diatoms. All use this one particular product as their storage product. They produce more keratin than the chlorophylls. They are now grouped with brown algae and water molds, and they include diatoms. These are components of marine phytoplankton. They are a major source of the world's oxygen. So there's a diatom. These are really pretty under the microscope. So there's some characteristics of various algae. You can read through this and admire it, but I'm not going to ask any questions about it because it's really not the focus of our course. Um, but there it is. Water molds. These differ from fungi in several ways. They have tubular cristae in their mitochondria. Cristae are the folds that you see on the mitochondria. So they're little tubes rather than folds. So their cell walls are made of cellulose instead of chitin. Their spores have two flagella, and they have two true diploid bodies rather than haploid bodies. Water molds decompose dead animals and return nutrients to the environment. Some species are pathogens of crops. In particular, the one that they list here, Phytophthora infestans, cause the Irish potato famine. Water mold also affects fish. And so if you've ever seen a fish get ick before, they get all fuzzy, that's what's causing it, is a water mold. A few other eukaryotes of microbial interest, including parasitic helminths and vectors. I'm going to talk about helminths later in the course. So parasitic worms, which are a big important issue in veterinary medicine, so I do talk about them later in the semester. They have microscopic infective and diagnostic stages. Arthropod vectors, these are going to be things like ticks and fleas. Again, important in vet med, so we'll be talking about them some more later. They are animals that carry pathogens, both as mechanical vectors and biological vectors. Disease vectors belong in two classes of arthropods, the arachnidia and the insecta. So some fun stuff to look at here. Right, so we're looking at some representative arthropod vectors. And again, these are of special concern in veterinary medicine. So arachnids, adult arachnids have 4 pairs of legs. The ticks are the most common important arachnid vector. Hard ticks are the most prominent tick vector. There's the soft ones that they call seed ticks as well, but the hard ones are the ones that cause the most diseases. And there are a few mite species that transmit rickettsial diseases that we'll be talking about. So this is where we're talking about ticks and mites. Technically spiders, but spiders don't really transmit diseases. They cause other problems. Insects, so adult insects have 3 pairs or 6 legs. So spiders and ticks and mites and all those have 8 legs, and insects technically have 6 legs, and they have 3 body regions. So this is where we're going to see fleas, lice, flies, mosquito. Mosquitoes are the most important arthropod vector of disease, especially for humans, but they can cause a lot of problems, as you probably already know, for animals as well, especially things like heartworm. And then kissing bugs. So I'll be coming back to all of the helminths and the arachnids and all that sort of stuff later in the semester. For now, make sure you have gone through your work in Pearson and take your quiz. And I will see you for the next chapter. Bye.